Human kidneys process an astonishing volume of blood every minute, driving an immense unseen liquid flow through millions of microscopic tubes. The sheer scale presents an immediate physiological problem. The kidney filters roughly 180 liters of blood per day, yet normal human urine output is only 1 to 2 liters. This means your entire blood volume passes through this filtration system approximately every 40 minutes. Without an aggressively efficient mechanism to reclaim this fluid, a person would die of profound dehydration in under an hour. The initial filtration of blood at the glomerulus relies on brute force physics. The process is governed by pressure differentials known as Starling forces. The primary driver is hydrostatic pressure. Fluid mechanics physically push blood plasma outward against the walls of the glomerular capillaries. Working against this is the oncotic pressure of the blood, a pulling pressure created by large dissolved proteins attempting to draw fluid back in. Because the outward hydrostatic pressure overwhelmingly exceeds the inward oncotic pressure, there is a massive net flow of fluid out of the blood and into the nephron. Reabsorption is a thermodynamic requirement dictated by the extreme pressure differentials at the filtration barrier. Once fluid enters the nephron, the first site of fluid reclamation is the proximal convoluted tubule, located in the outer cortical layer of the kidney. The proximal tubule performs bulk transport, reabsorbing about 70% of the filtered sodium, amino acids, and water back into the body. However, this mechanism is limited by its isotonic nature. Because it reabsorbs water and salt in equal proportions, it cannot concentrate the remaining waste. To truly conserve water, the kidney must decouple the movement of water from the movement of salt. Achieving this requires a specialized architecture. Bulk reabsorption only delays dehydration. Ultimate survival requires a strict spatial gradient that dives deep into the organ. This spatial engineering occurs in the loop of Henle, a U-shaped fluidic circuit diving downward from the cortex deep into the kidney's inner medulla. The descending limb's membrane is permeable to water, but impermeable to salt. The surrounding deep medulla tissue is hypertonic, possessing a high solute concentration. As filtrate travels deeper, osmotic pressure rapidly pulls pure water out of the descending limb to be reclaimed. Water only passively exits the descending limb because the surrounding tissue is actively pulling it out. This raises the question of how the medullary tissue became so concentrated in the first place. The answer lies in the ascending limb of the loop of Henle, as the fluid turns the corner and heads back up toward the cortex. Here, the structural role is inverted. The ascending limb is impermeable to water, but highly permeable to salts. Packed with mitochondria, the thick ascending limb provides ATP to actively pump sodium and chloride into the medullary tissue. This generates a multiplier effect. Pumping salt out on the way up creates the hypertonic gradient that pulls water out on the way down. Adjacent blood vessels, flowing in the opposite direction, capture the reclaimed water and salt without destroying the gradient itself. The nephron functions as a coupled thermodynamic engine. The active metabolic work of the ascending limb drives the passive water conservation of the descending limb. To test how reliant this engine is on its precise geometry, we can examine a specific genetic anomaly: mice engineered to lack the enzyme carbonic anhydrase 2, or CA2. Researchers observed two distinct behavioral phenotypes in these CA2 deficient mice: polydipsia, or excessive drinking, and polyuria, or excessive urination. This bar chart shows that the urine of the knockout mice is significantly more dilute than that of the wild-type controls. Producing dilute urine indicates that these mice have lost the ability to concentrate waste, suggesting a failure in their water reclamation hardware. While the urine volume indicates the body is losing water, locating the specific failure point requires interrogating internal regional data from the kidney itself. The experimental hypothesis suggests that CAII acts as a catalyst to activate specific cellular water channels called aquaporin 1. This visualization maps internal chemistry across 3 zones: the cortex, the outer boundary isome, and the deep inner medulla, to track failing aquaporin channels. While outer regions remained normal, the deep inner medulla of the knockout mice suffered a sharp drop in osmolarity and sodium concentration. Aquaporin 1 channels are primarily located in the thin descending limb, deep in the medulla. This regional drop identifies the specific location of the failure. Identifying the location reveals a contradiction. If water fails to exit the tubule and remains trapped inside, the tissue on the outside should theoretically be more concentrated, yet the data shows a loss of solutes. If water fails to exit the descending limb, it should not dilute the surrounding tissue. In an isolated system, the tissue concentration would actually increase as less water enters the space. View the loop of Henle as a coupled system. In deficient mice, inactive aquaporin channels keep water trapped in the descending limb. This dilute volume enters the ascending limb, starving ion pumps of sodium, which collapses the hypertonic tissue gradient seen in clinical data. A failure of water permeability upstream systematically starves the ion pumps downstream, which in turn destroys the entire concentration gradient. Biological systems are defined by these sequential downstream consequences. In the kidney, trapped water doesn't just stay in the tube, it compromises the concentration engine. Biological equilibrium requires continuous, microscopic thermodynamic precision to maintain fluid balance. The kidney's functional power is a product of its spatial geometry, relying on the physical decoupling of salt and water to sustain the organism. The countercurrent multiplier is a life-saving engine, providing physiological stability for survival outside the water.